Okay, so we're going to go through some more properties of macromolecules. So we're going to review carbohydrates. Remember, the monomer is monosaccharides, the polymer is polysaccharides, the bond name is glycosidic bond, the function, short-term energy storage, the structure, carbon, hydrogen, oxygen in a 1:2:1 ratio, and it can also be linear or ring-shaped or branched. Okay, and we have three main examples: starch, cellulose, and glycogen. So plants and carbohydrates, they are going to, plants use both starch and cellulose. So starch they're going to use for the energy storage. For plants, you can usually think of like starchy plants that's storing energy. Okay, and then the cellulose is going to be the structure in the cell wall. So where would the cell get the monomers from? For both of these, the monomer is glucose because plants are going to be able to make glucose from photosynthesis. Okay, so cellulose is used in the rigid cell walls of plants. It offers support and protection for the plants. But humans, we lack the enzymes needed to break down this bond. So remember that bond is called a glycosidic bond. So that bond in between the monomers of our carbohydrates of cellulose, we cannot digest. Herbivores can, and that's actually not because they themselves have enzymes that can break down this bond, but they have bacteria in their guts. So cows, for instance, have this bacteria in their guts that the bacteria is able to break down that cellulose so that they can digest it. Glucose, the reason starch and cellulose are so different, they're made of glucose, yes, but one is made of alpha glucose. We have alpha glucose and beta glucose. So alpha glucose and beta glucose, notice the only difference in their structure is right here. And they're made of the same elements. It's just switched the direction. So that little change is what's going to be the difference. Okay, so starch is made up of all alpha glucoses, but cellulose, on the other hand, is made up of alpha and beta glucose that alternate. So here's an example. We have an alpha glucose, alpha glucose, alpha glucose, alpha glucose. But cellulose, we have alpha, beta, alpha, beta. So that makes this bond just not recognized by the enzymes. So here, that glycosidic bond looks different because of that change to a beta glucose. So we can break and digest with enzymes, but this is not recognized by the enzymes. The enzyme does not recognize it, and we just move on to the next molecule. So what's the process called? Remember, when we break the bonds of polymers in general, it's called hydrolysis. So we're requiring water here. So every time we do this reaction, the enzyme will use a water molecule for this process. And then we'll end up with our monomers of glucose that can be used. Glycogen. Animals can digest glycogen because it is made of all alpha glucose monomers, similarly to starch. Just the big difference is it's very branching structure. So it's used for fast energy storage in animals. It's usually stored in the liver and muscle cells so that animals can quickly, including humans, can quickly get those glucose monomers. Okay, now we're going to shift focus to lipids. Remember, the monomer is glycerol and fatty acids. The polymer is lipids. Function is long-term energy storage and structure, like in membranes. And its structure is carbon, hydrogen, oxygen, and sometimes phosphorus. So here we have the structure of a lipid, a triglyceride actually, because it's got 3 here. And we have that glycerol on the back. And then a phospholipid will have that phosphate group making a hydrophilic head and those hydrophobic tails. Okay, so a fat molecule versus a phospholipid, they look very similar. They're going to have a glycerol backbone and we're going to have fatty acid tails. Here, the big difference is the presence of that phosphate or not. So fat molecule and a phospholipid. The fat molecule is 100% hydrophobic. We can see that by seeing that it's made of pretty much entirely carbons and hydrogens. There's a few oxygens scattered here, but for the most part, it's carbon and hydrogens. Our phospholipid is going to be hydrophobic over here and then hydrophilic up here where that phosphate is. And you can really see that because of that negative charge on that phosphate molecule. We call these amphipathic. That's the word for when a molecule has both hydrophobic and hydrophilic tendencies. Steroids are also going to be considered lipids. They're usually 4 fused rings, something like this. Remember that each of these turns in these ring structures is carbons and hydrogens. So it's made of almost all carbons and hydrogens. There's one little oxygen over there. And this doesn't look anything like our other lipids we've been talking about. The only reason steroids are classified as a lipid is because they are hydrophobic because of that primarily carbon and hydrogen structure. Okay, shifting gears to proteins. Remember, the monomer is amino acid, the polymer is polypeptide, the bond name is a peptide bond. The function has all the different functions, and enzymes are the main function. And then the structure is carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Really, they all have sulfur, just a very small amount. And then they have that amino group, alpha carbon, carboxyl group, and an R group. Okay, so proteins can make really diverse structures, which we've mentioned before. They do need to be folded properly, and there are 4 levels of protein folding and protein structure: primary, secondary, tertiary, and quaternary. So the primary structure of proteins, this is just the basic sequence of amino acids. And remember, these different amino acids could be hydrophobic or hydrophilic, polar or nonpolar, another way of saying that. And so we have them, and they can be in different orders. So just that sequence, that order, that's the primary structure. They are also directional. So we have an amino terminus, an amino end, and a carboxyl terminus or carboxyl end. So the big thing you're looking for is nitrogen, carbon, carbon. Nitrogen, there's going to be a carbon there. Carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. And notice that means at one end we have a nitrogen, the amino, and one group we have a carboxyl, that carbon. Okay, so they're always going to be synthesized from N terminus to C terminus, so N to C. So here we have our N end, and then these are just different amino acid names, and then at the other end we have the C. So when we're synthesizing new amino acids or new polypeptides, we're going to add amino acids from that N to C direction. So if we were to add more onto here, they would go on this end of our growing polypeptide chain. So that is our primary structure. Our secondary structure is alpha helices and beta sheets. These are going to be like little spirals. It's the alpha helix, and the beta sheets are where we have different amino acids kind of lining up together. The big thing here is that they're held together by the hydrogen bonds in that backbone of the polypeptide, of the peptide, and not the R groups in themselves. This structure won't come up as much this year, but that's the secondary structure. Here's another example. The beta sheets or beta pleated sheets. The tertiary structure is one of our really important structures. This is the 3D structure that's going to determine the function of that protein, and they are determined by the interactions of those polar and nonpolar R groups. So this is probably the most important for our folding of our protein. So here is an example. We have that string with our of our backbone, so like NCC, NCC, you can think of that happening here. And then these little circles are representing the R groups attached to it. So here we have red representing hydrophobic regions and blue representing hydrophilic R groups. And so because of that, this is what's going to drive the folding of our protein to fold it properly. Okay, so think about where would those polar R groups be? So those polar R groups are going to want to be near water, and the nonpolar will want to be away from the water. So they're going to fold so that those hydrophobic regions are inside, protected and surrounded by our hydrophilic regions. Okay, so that's our tertiary structure, probably our most important for structure and function of our protein. Quaternary structure, this is basically when we have multiple subunits of proteins coming together. A great example of this is hemoglobin. Hemoglobin is made of 4 different subunits that come together to make an overall superstructure. Okay, so that is our quaternary structure. It won't come up so much in this class. Okay, now we're going to shift gears for our nucleic acids. So nucleic acids, remember our monomer is a nucleotide, our polymer, DNA and RNA. The bond name is a phosphodiester bond, and its function is for storing that genetic information. The structure is carbon, hydrogen, oxygen, nitrogen, and phosphorus, and it's going to have a 5-carbon sugar, a phosphate group, and a nitrogen base. Okay, so just like with other macromolecules, if we change something in its structure, it's going to change its function. So we have our DNA nucleotides. If we change that sequence, it's going to change the information that's encoded. It will change the function. Okay, we have 2 different types of nucleic acids. We have purines and we have pyrimidines, and this is talking about the structure of those nitrogen bases here. So the structure here. So purines have 2 rings in their structures. And pyrimidines, have 1 ring in their structures. So the way I remember this is pure as 2 gold rings. So purines have 2 rings, and they're pure as gold. So adenine and guanine, both are purines. All the other ones, cytosine, thymine, and uracil, are examples of pyrimidines. The reason that this is important is because whenever we have the nucleotides bonding together with hydrogen bonds, it's always a purine bonding with a pyrimidine. So adenine binds with thymine and guanine binds with cytosine. So notice that's always a purine and a pyrimidine. It's also important because if we have a mutation, it's sometimes easier for a mutation to happen and not get corrected if like one purine is substituted for another purine, or one pyrimidine is substituted for another pyrimidine. So sometimes those errors happen. They're called mutations. A lot of times the cell has things in it that catch those mistakes and fix them. But if there's something like an adenine instead of a guanine, they are very similar in structure. So sometimes those mistakes go unfixed. Nucleic acids, just like proteins, are also directional. So they have a 5' end and a 3' end. The 5' is a phosphate end, and the 3' is a hydroxy end. So I remember 5 to 3. And I remember this actually in conjunction with our proteins being directional as well, and because it kind of rhymes, because proteins go N to C, and this is 5 to 3. So N to C and 5 to 3. If you had them the other way, they wouldn't rhyme. So here we have our basic shape of a nucleic acid. And you can see we have a 5' end and a 3' end, and these are where they're attached. So you can see here 5' end, 3' end, and that's where it attaches to the next thing, the 5' end and 3' end. And then our nitrogen bases are sticking out here, kind of in the middle. Okay, when we have double-stranded, like in DNA, it has 2 strands. And you might notice about these strands, one end is 5', and then the 3' over here, and this one, the 5' is here, and the 3' is up there. We call this anti-parallel direction. So they're parallel because they're going in the same direction, but they're anti because one is 5 to 3, and the other is going the opposite direction. This is really helpful, especially when we get to things later in the year, because if we know that one side is 5, then the other one has to be 3. So that will come up later. The replication occurs also in this direction, 5' to 3'. So from here, 5 to 3, it would replicate in this direction. If we were adding to this other strand, it would go 5 to 3 in this direction. So it's always synthesized 5 to 3. Okay, another important thing about our DNA is that the way that our base pairs that bond together are connected is through hydrogen bonds, the same type of bonds that hold together our water molecules. So it's not like an actual bond, but it is very strong because we have so many of these hydrogen bonds occurring. Okay, and you might notice too here between them, there's actually a different number when we look at our adenine and thymine than our guanine and cytosine. So adenine and thymine, whenever they're bonded together, it's 2 hydrogen bonds, and guanine and cytosine is comprised, they're connected with 3 hydrogen bonds. So that means our guanine and cytosines are held together a little stronger, and so that will come to play throughout the year as well. Okay, that is it for today. Hope this is helpful.